main divisions of the nervous system?
Central and peripheral.
Very good, central and peripheral, there we go. Okay, so I know you've all heard this terminology before. I think we hear of the central nervous system a lot because it's kind of the, the structure that really controls everything else in the body, of course, along with the peripheral nervous system, but we hear so much about the brain and the spinal cord, so I think everyone's familiar with that. So the two main divisions are the central nervous system, named central because it's centrally located. This is the brain and the spinal cord, and they are, of course, connected to each other, abbreviated CNS. And then the peripheral nervous system, abbreviated PNS, would be all of the nerves that branch off of the brain and the spinal cord. And these go out into the periphery of the body. They go to almost every single tissue in the body. There's very few tissues, like maybe parts of the tooth, that don't have nerve supply. Just about every tissue in the body has nerve supply. So depending on whether these are connected to the brain or to the spinal cord, we call these two sets of peripheral nerves, cranial nerves or spinal nerves. Okay, and again, cranial branch off of the brain, and spinal nerves branch off of the spinal cord. And these go out into all the organs and tissues, and that is what makes up the PNS. Okay, and taking a look at this, here it is. Okay, so again, brain and spinal cord make up the central nervous system, and all these nerves going out into all the tissues are the peripheral. Alrighty, so you can see everything is connected. All these different parts of the nervous system are all connected to each other. Okay, so we know that we have two types of information that are carried on our nerves, right? Sensory and motor. So let's talk a little bit more about this. And this is for now just an introduction to these terms. Okay, so if this doesn't make a lot of sense to you now, don't worry about it. As we go into the nervous system, this will make more sense. So when we talk about sensory information, we have sensory information, of course, that's coming into the central nervous system, but from different parts of the body, right? That makes sense. So this information is divided into two groups, either somatic or what is called visceral. Okay, the term soma or somatic actually just means body. So this doesn't really tell you a lot about where it's coming from, but this is sensory information from the skin, the muscles, and joints. So specific types of sensory information only from those areas. Now when you hear the term viscera, if you guys are familiar with that term, or visceral, this refers to our internal organs. Okay, so this is sensory information from the internal organs. So sensations like cramping, or nausea, or fullness, hunger, those are the types of sensations that these nerves would be carrying. Okay, and then same thing for motor neurons. We know motor neurons carry action potentials out to our muscle and to glands. We've talked about those a little bit already. There's two types of motor neurons. Okay, so we have, again, a group called somatic, and somatic motor neurons are actually what we've already talked about. I didn't use this term, but they would be this group that we've already talked about. So these would be motor neurons going to skeletal muscle. It kind of makes sense. These would be in a separate grouping because you guys know skeletal muscle is the only type of muscle we can control voluntarily, right? Yes. Okay, so notice that there's a different term here for the other group of motor neurons, autonomic. Autonomic is sometimes referred to as visceral. And so again, when you think about autonomic, think about internal organs. Okay, so we know that in the internal organs, we have what type of muscle? the other two types, right? So cardiac in the heart and smooth muscle. So these would be motor neurons that go to those two types of muscle tissue. Okay, so to cardiac and smooth muscle. Again, this makes sense because we know these are gonna be different than motor neurons controlling skeletal muscle because these two types of muscle tissue are not controlled voluntarily. Okay, and also located in our organs, glands, right? Remember the definition of a motor neuron is a neuron that carries the action potential to muscle or glands. And so again, things we can't control voluntarily are controlled by these autonomic or visceral motor neurons. Okay, so just a little bit of terminology for now, and we'll come back to those terms later. Questions? Okay. And then you have a table in your book that shows you pretty much exactly what we just talked about, okay? This is what we just talked about. Moving on. Okay, so let's talk about nervous tissue. You guys know in the body we have 4 categories of tissues, right? Epithelial, connective, muscle, and nervous tissue. Nervous tissue is in a category of its own because it is totally different from everything else, a very unique type of tissue. And of course, nervous tissue is located in the nervous system, right? Brain, spinal cord, and our nerves. Okay, so in nervous tissue, we have 2 types of cells. First, we're gonna talk about neurons. These are the ones we've talked about a little bit already. The other group of cells is what's called neuroglia, and they're kind of like supporting cells. They support neurons in different ways. So let's first talk about neurons. These are also called nerve cells. You guys know that neurons communicate with each other, also control muscle contraction, also control secretion of glands by sending these electrical signals that we call action potentials. This is how neurons do everything. It's how they receive sensory information. It's how we think when we're thinking about something and we're making a decision. We're storing memories. We're seeing or hearing or listening or tasting, whatever sensory information happens to be coming in. It's how we decide when and how we're going to move. Everything is done with action potentials. Okay, interesting thing about neurons is that once the nervous system is matured, almost all of our neurons are incapable of dividing by mitosis. So we kind of are stuck with what we have. We can't really do much to increase the number. There are a couple areas in the brain where neurons do divide, and fortunately one of those is in the hippocampus, which is important for memory. But most of our neurons can't divide once they're mature. That is why when we damage parts of the nervous system, like when a nerve is severed or a part of the brain is damaged, like with a stroke or whatever it happens to be, there is often loss of function. Not always, but that happens because the neurons do not always heal well and do not divide to replace, the damaged neurons. So let's talk about the different types. There's 3 types based on their structure and also 3 types based on their function. Okay. Alrighty, so first of all, 3 types based on their function. Now we know we have sensory and motor. Remember that the definition of a sensory neuron is a neuron that carries impulses, and again, this is just another word for action potentials, okay? So don't let that word scare you, it's the same thing. From the sensory receptors to the CNS. So these would be incoming, right? Incoming signals coming in from, could be sensory receptors in the skin, it could be in our muscles and joints, it could be in organs, And it could be information that's, coming in from outside, like what we're seeing, hearing, tasting, smelling, etc. Notice that this is another word for sensory, so you can always say afferent information when you're talking about sensory information. You can also say afferent neuron instead of sensory neuron. Okay, motor is also referred to as efferent, okay? We already know that motor neurons are the neurons that carry action potentials, okay, or impulses from the CNS to muscles or glands. Okay, so again, these are the actual definitions of these neurons. Now, it seems like that would be it, right? Either a neuron is sensory or motor. But in the central nervous system, in the brain and spinal cord, we actually have billions and billions of another type of neuron. And these are called interneurons, also referred to as association neurons. Very interesting because you don't really hear much about them. You don't really hear about interneurons, in your daily life. But these by far outnumber the other two types. We actually have over 99% of all of our neurons are this type. Because you can imagine how many neurons we have in the brain alone. Billions and billions of these, connecting different areas and allowing them to communicate with each other. So sometimes these are longer, like it could go, from the frontal lobe back to the occipital, or even from the brain down into the spinal cord, okay, connecting the brain and spinal cord. As long as they lie entirely within the CNS, it is called an interneuron. So again, some of these are longer, but some are going to be very short, just connecting areas that are right next to each other in the brain. And so this is what allows parts of the brain to communicate, allows, the brain and spinal cord to communicate. So let's talk about what these neurons look like. You guys already know what motor neurons look like, right? Talked about that a little bit already. Okay, so any questions before we go on? Okay, so there's a diagram that shows these that we'll take a look at in just a second. And that diagram will actually be on the test. Okay, so neurons are named after the number of processes, or those things that stick out from the cell body. They're called processes. They're named after the number of processes attached to the cell body. So this is the type that we have already talked about because these actually are the motor neurons. This is what they look like. So multi, of course, meaning many. Multipolar neurons have many dendrites and one axon. So we drew these before. We know that all these things connected to the cell body that receive the incoming signals, right? Those are called dendrites. Okay, so multipolar neurons have many dendrites. Receive incoming. It is estimated that neurons in the brain have approximately, each one has approximately 1,000 synapses, or on average about 1,000 synapses with other neurons. So there's a lot of incoming signals all the time. And so based on all the incoming information, if this neuron decides to send an action potential, it will send it along this process. What is this one called? The axon. We talked about how axons usually branch many times. So we'll put some branches here. And remember, at the ends of the axon branches, we have these little knot-like structures called synaptic nodes or synaptic bulbs, exactly, with the little vesicles. What do the vesicles contain? Acetylcholine, or it could be another neurotransmitter. They contain a neurotransmitter. So when the action potential gets down to the end of the axon, the neurotransmitter is released. Okay, so here's our axon, and these only have one axon, and this will send the action potential. The action potential usually begins right here at the beginning of the axon and it will travel the entire length of the axon, and then the neurotransmitter is released. Okay, so we know that motor neurons look like this, but also those interneurons we were just talking about look like that also. They are multipolar. So this type of neuron, multipolar, are the most common because all of our interneurons are multipolar. This next type we're going to talk about is actually the least common. Bipolar would have how many processes attached to the cell body? Two, right. So one of these will be the dendrite and the other will be the axon. And we'll take a look at them in just a second. These are found only in the special senses. Okay, so here the special senses for these neurons includes just the nasal cavities, okay, where they're going to carry smell, the retina of the eye, and the inner ear, where they would carry both sound and equilibrium. Okay? Oh, inner eye. Oh. I don't know. What's going on? Okay, inner ear. Okay, and then the last type of neuron is a unipolar, uni meaning one. So there is one short process attached to the cell body that splits. Okay, and we will take a look at that in just a second. These are our other sensory neurons, okay, besides the special senses. So all of our sensory neurons that carry like touch, pressure, pain, temperature types of sensations are going to look like this. They're unipolar. Okay, let's take a look. Okay, so here are the three types. This diagram will be on the test. I'd like you to be able to identify the type of neuron based on its structure and be able to identify these processes, okay? So notice there's a couple different multipolar neurons here. These are the same. They're just showing you they may not always look exactly alike. This one has more branching in its dendrites. Doesn't matter. It's the same. Okay, so a bunch of dendrites, one axon. You can't see enough detail here to see the synaptic knobs, but they would be here at the ends of these little axon terminals, the ends of the axon, okay? So these would be synaptic knobs with the neurotransmitter. This is a bipolar neuron. Notice how this process carries the signals to the cell body. So this is the dendrite, and this would carry the action potentials away from the cell body. So this is the axon. Again, you would have little synaptic knobs here, we just can't see them, okay? And then finally, the other type of sensory neuron, the unipolar, notice one process attached to the cell body, but it's going to be very short, and then it will split, okay? So the dendrites in this type of neuron are only right here. They're, going to be out in the periphery of the body somewhere, like in the organ or in the skin, wherever it happens to be. This is the beginning of the axon, right here. Okay, so this whole process is the axon. You can just call this whole thing the axon. And again, there'd be little synaptic knobs here with the neurotransmitter. Now, because this part of the axon comes in from the periphery of the body, and it comes into the cell body of the neuron, this can be called the peripheral process. And this will then carry that information into the central nervous system. So you can call this part of it the central process, but you don't have to. It's easier, I think, just to call the whole thing the axon, and that is perfectly correct. Okay? You guys have any questions on that? Okay, we do have a multipolar neuron model. And so we're going to take a look at a multipolar neuron kind of a little bit more up close and talk about just a few things that neurons have that other cells, or that are kind of different from other cells. And you can also see these structures we're going to talk about. You can see them on this model. Okay, this is, just so you know, You guys know this is the cell body. So these things that come out from the cell body, these are the dendrites of this neuron. These are axons of other neurons that are synapsing on this one. And this is just the axon kind of blown up, so you can see it up close. Okay. So notice here, again, this is the axon. You can see the synaptic knobs here, and these are all dendrites. They made the newer additions a little bit more detailed. You can see these little spines coming off the dendrites. They're called dendritic spines. I don't have them on this diagram, but you guys can probably see them in your picture. It just adds some surface area to the dendrites, making them more sensitive. That's what the dendritic spines do. Okay, so let's take a look here at the beginning of the axon. Notice there's this kind of like funnel-shaped region where the axon connects to the cell body. So again, the whole thing here is the axon. So this part of the axon is called the axon hillock. This is the most sensitive part of the axon electrically. So this is usually where the action potential will begin, right here at the axon hillock, and then it will travel the entire length of the axon. Okay, so the action potential begins here. And we'll come back to these things that are wrapped around the axon later. This is actually a type of neuroglial cell, so we'll talk about those later. Okay, so you can see here in the cell body, notice there's a bunch of these little splotchy structures that kind of look dark. You can actually see these if you look at the neurons in the microscopes. You'll see these. These are called either, you can say chromatophilic substance. Chromatophilic means color-loving, because these will pick up stain, and so they stain dark. That's where that name comes from. Or you can say Nissl bodies or Nissl substance, which I think is easier. These are named after someone who studied neurons. And what this is actually rough endoplasmic reticulum and free-floating ribosomes. So you guys know that rough ER has ribosomes attached, right? So neurons have a bunch of ribosomes. What do ribosomes do? You guys remember, I know you do. Protein synthesis, right? It's the site of protein synthesis. The reason why neurons have a lot of ribosomes is because the action potentials travel along the cell membrane. So their membrane tends to wear out over time. And so they're always replacing parts of the membrane, and so that is why they have a lot of rough endoplasmic reticulum and free-floating ribosomes. Okay, and that's what that splotchy looking stuff in there is. Okay, also, something that's not really shown in this picture, but you'll see this in other diagrams, and also on the model shows this. There's these kind of fine lines. Oh, this is black too. Oh well. You'll see these kind of fine lines that come out from the cell body and go into all the dendrites and down into the axon. And what these are is actually, it's really part of the cytoskeleton. You guys know the cytoskeleton holds the shape of the cell. So these are important in neurons because, as you can imagine, if the neuron loses its shape, it's not going to be able to form synapses with other neurons. So these are very important. They are called neurofilaments. They are actually intermediate filaments, which again are just part of the cytoskeleton. And these tend to form kind of bundles that are called neurofibrils. And so this is kind of like myofilaments are bundled into myofibrils, right? If you remember from muscle tissue. So you can use either name, I don't care. So these just help to maintain the shape of the cell. Mm-hmm.
Since 14.3 isn't all that, this that you gave us, do you want us to just know the stuff that you're writing down right now? Or, since the
Yeah.
part of the model?
So you guys remember that although I give you figure numbers for the test, remember that you still want to study these other diagrams. When you're studying, you know, the parts of the whatever you're studying, you always want to study the other diagrams because you still have to know these structures that we're talking about. But I will ask you to identify them on this model, okay? And you can see the neurofilaments or neurofibrils here. They're shown in these dark lines. Okay, all this blue stuff in here would be the Nissl bodies. Okay. You guys have any other questions on any of that? Okay, so let's talk a little bit more about synapses. And since we've already talked about the neuromuscular junction, you guys kind of already know what the synapse is going to look like. This is actually going to be exactly like the neuromuscular junction, except instead of a muscle cell membrane, we'll see that there is a membrane of the next neuron. So this is very common terminology when you're talking about a synapse. And because when you guys take physiology, you'll talk about synapses a lot. You will be using this terminology all the time when you take physiology. Okay, so it's just a way of identifying the two neurons, the pre and the postsynaptic. Okay, so let's take a look. You can ignore this because this is a gap junction. Okay, they're just showing you this so you can compare it to a synapse, but you can ignore that. Okay, so this is the end of our axon, right? This is a synaptic knob or synaptic bulb. And notice there's a little synaptic vesicles with the neurotransmitter here. They are actually using acetylcholine as the neurotransmitter. Something you are familiar with. Okay, so we got our little acetylcholine molecules in our synaptic vesicles. Now when the action potential, or again, same thing as the nerve impulse, I'll just abbreviate action potential here. When it reaches the end of the axon, we know that the neurotransmitter will be released and it's going to travel across this little microscopic gap. What is that gap called? Synaptic cleft, exactly. So just like in the neuromuscular junction, there will be a synaptic cleft. The neurotransmitter will diffuse across that and bind to receptors on the next neuron's membrane. So this is exactly like the neuromuscular junction, except this is not a muscle cell membrane, it's the membrane of the next neuron. And you can see it has receptors for acetylcholine. If enough acetylcholine is received, then this neuron may have its own action potential, okay? So again, the binding of the neurotransmitter is what can trigger an action potential in that neuron or muscle fiber, whatever it happens to be, okay? So that is the structure of a synapse. So where can synapses take place? Like where are they located on a neuron? Well, actually they can be anywhere. So let's take a look at the different possibilities. If the axon of the presynaptic neuron synapses on dendrites of the postsynaptic neuron, then it's called an axodendritic. So this name just tells you it's between the axon and dendrites. Here it would be between the axon and the cell body. Remember, soma means body. And here it's between the two axons, okay? And you can see these different types of synapses on this model. We do not actually have an axoaxonic on the model, but you can see this one right here would be an axosomatic, and these where you see them on the dendrites here would be an axodendritic, okay? Okay, so this diagram shows you what that would look like. Notice we have, what type of neuron would this be based on its structure? Multipolar, right, so we have two multipolar neurons. This is just a simplified way of drawing a multipolar neuron, so you don't have to keep drawing all those dendrites. They're just drawn like this. Okay, so it's just a simplified version. So notice here in this diagram, we have all those different types of synapses, where this presynaptic is synapsing on the cell body here. This would be an axosomatic. This would be an axodendritic. And then finally, these are not very common, but an axonic. Okay? So they can happen pretty much anywhere on that postsynaptic neuron. Okay, so I think that's about it as far as neurons. Oh, and then this next diagram actually just shows you kind of how these would be organized or laid out in the body, in case you're wondering, like, what would this look like in real life? So this is just showing you a sensory neuron carrying information into the spinal cord. Here's our spinal cord. And notice that now this sensory information that's coming in could trigger some kind of a response. This is really what our reflexes look like. So say you have something irritating your skin, right? This is information coming here from the skin, something that's itchy or biting you or whatever. So the sensory neuron, notice it is unipolar, okay? This is what our sensory neurons look like. And so this axon is carrying the action potential into the spinal cord. Notice here we have a little short interneuron that's connecting the sensory and the motor. This interneuron is what type based on its structure? Multipolar, right, multipolar. And that's what interneurons look like. They are all multipolar. And then here we have a motor neuron, again, multipolar. And the axon is going to go all the way out to muscles that we would use to, have a reaction to that, whatever, that's irritating the skin. So this really is a reflex, is what they're showing. Notice how long these neurons are. They are really this long. These are the longest cells in the body. Neurons are the longest cells in the body because these could literally go, the longest would be like from the brainstem all the way down into the foot. So many, many feet long sometimes. So this sensory neuron comes all the way from the skin here in this area and goes all the way into the spinal cord. And same thing with the motor neuron, it's going to go all the way from the spinal cord out to the muscles, okay, wherever they happen to be. And they travel in nerves. So as they're going out to the body part, they would go in a nerve. That's how they go, kind of out to the periphery from the central nervous system. Okay? Does that kind of make a little sense? Do you have any questions on any of that? Okay, so that is it for neurons. The other type of cell in nervous tissue is what is called neuroglia, or you can just say glia or glial cells. And these are kind of supporting cells. They have different functions. They support neurons in different ways. They are located in both the central and peripheral nervous system. These are much smaller than neurons, but they outnumber neurons by a lot, like about 50 to 1. And these cells can divide. So these are the cells that when people have brain tumors, they are usually tumors of neuroglial cells because neurons can't divide to form tumors. So brain tumors are usually a type of neuroglia. These divide throughout our lifetime. Okay, so we're going to talk first about the 4 types of neuroglial cells in the CNS, and then we have 2 in the PNS. Okay, so this is just showing kind of a cut through the brain. This is a frontal section through the brain. They're just showing a little kind of section of tissue blown up, so you can kind of see what this might look like with all these different neuroglial cells around the neurons. Notice here we have 2 multipolar neurons and a bunch of different neuroglial cells around them, okay? So there are 4 types in the CNS. Let's talk about astrocytes first. Astro means star-shaped. So these cells have a lot of processes that travel out from the cell body, and they connect to neurons. They also wrap around blood vessels to form what we call the blood-brain barrier. So astrocytes have different functions. This is just a table that shows you all these different neuroglial cells, what they look like and what their functions are. So this just gives you kind of a quick reference. Okay, so astrocytes, again, they have these processes that reach out and wrap around parts of neurons that are nearby. So they structurally support the neurons, kind of hold them in place. They also wrap around capillaries. Capillaries are the smallest blood vessels in the body. So just say they wrap around these capillaries, forming what's called the blood-brain barrier, which I'm sure you've all heard of. And this just gives an extra layer of protection around the blood vessels. So if we have some kind of sudden change in the blood chemistry, like say a toxin has gotten into the blood, this will hopefully prevent it from getting into the brain, which could be fatal very quickly. But this does also limit a lot of medications from being used in the central nervous system, because some medications can't cross over the blood-brain barrier. Fortunately, caffeine can. Okay. Also, these cells will form scar tissue if neurons are damaged. Again, neurons can't really divide to replace, injured cells, so they're often replaced with scar tissue. And they help to also maintain the normal electrolyte or ion levels in the fluid, the interstitial fluid around the neurons. This is important because this is actually how action potentials are sent. Action potentials are where the electrolytes or ions cross over the cell membrane. So if these are kind of out of their normal balance, then that can affect the action potential. Okay, moving on. And this is what an astrocyte might look like. You can see some of its processes kind of attaching to the neuron, and others would wrap around this nearby blood vessel. Okay, so the next type of neuroglial cell, ependymal cells. Ependymal cells actually line cavities that are in the brain that are called ventricles. Okay, and so this is really a type of epithelium. You guys remember that epithelial tissue lines all cavities. It is simple cuboidal with cilia. And so we have this lining, not only the ventricles in the brain, these spaces in the brain, but also there's this narrow canal that goes down the spinal cord, and this tissue will line the central canal of the spinal cord. Okay, so in certain parts of the ventricle, there is a structure called the choroid plexus. And we'll talk more about this next time when we talk about the ventricles, but what happens in the choroid plexus is this is where cerebrospinal fluid is formed, okay? So it actually comes from the choroid plexus in each ventricle. So I'm just going to say located in the ventricles. So these cells will line the ventricles. They also make cerebrospinal fluid, and then the cilia moves the cerebrospinal fluid along through the ventricles, so it keeps it moving and circulating. kind of what ependymal cells look like. This would be the cavity of the ventricle or the central canal. And here is the cilia, which would be moving the cerebrospinal fluid along. So all of this fluid here is cerebrospinal fluid. Okay. This would be cerebrospinal fluid. And this would be the tissue of the brain or spinal cord. Okay, so the next type of neuroglial cell is called microglia. These are phagocytic, meaning these will ingest and destroy foreign invaders. So this would be like the macrophages in our connective tissues that wander around looking for, pathogens or foreign invaders. And they look kind of like this.
Is it possible for those to be destroyed as well, or no?
I'm sure that, yeah, they die,
Oh,
and
they can't replicate, or?
yes, they can. Yeah, these neuroglial cells divide. They normally divide throughout our lifetime. Okay, and then finally, the last type of neuroglial cell in the CNS is called an oligodendrocyte. Long name, and what they do is they form a fatty substance called myelin. Myelin electrically insulates the axons from each other, so that while the axon is sending an action potential, it doesn't automatically get transmitted to all of the surrounding axons. Because remember, the action potential is electrical current. So this would be like a wire that doesn't have insulation. That electrical current will then kind of get sent to all of the surrounding structures. So we don't want that to happen. And so myelin wraps around the axon, electrically insulating it. This will also affect how the action potential travels. And we'll talk more about that in a little while. This is also what gives us white matter. Myelin is what gives us white matter. You guys have heard of white matter and gray matter? Well, the myelinated axons is actually what white matter is. They make up the white matter. This is also what breaks down in the disease multiple sclerosis. It will degenerate, and so people who have MS have sensory and motor symptoms depending on which neurons are involved. Okay, and this is an oligodendrocyte here. This is a cell body, and you can see these cells also have these processes that reach out and they wrap around the nearby axons, okay, forming what's called the myelin sheath. So they're forming the myelin sheath here by wrapping around the axon. Mm-hmm.
Do we need to know how the myelin sheath is made, or no?
Yeah, we're going to talk about that in just a few minutes.
Oh,
Okay, so these are the types in the CNS. Okay, so let's talk about the two types in the peripheral nervous system. There's only these two types of neuroglial cells in the PNS. First of all, satellite cells surround neuron cell bodies in the peripheral nervous system. And so they not only kind of protect the neuron cell bodies, but they provide nutrients to them. Okay, and we will take a look at that. A ganglion is just a grouping of neuron cell bodies. Okay, the grouping of neuron cell bodies out in the peripheral nervous system. Oops, peripheral. And this is how neurons are organized. So they have their cell bodies located in kind of little groups, and then their axons travel together. Ganglia is plural, ganglion is singular, just so you know. It's basically the same thing. Okay, so notice here we have some neurons, and this is a little grouping of neuron cell bodies, so this is a ganglion somewhere, okay, out in the PNS, this is a ganglion. Okay, what type of neurons are these based on their structure? Where they have the one process attached to the cell body. This would be the nucleus, and then that process splits. This is what they look like. So these would be unipolar, right? One process attached to the cell body. Yeah, so they're unipolar. And you can see these pink cells that are kind of like all around the cell body. These are the satellite cells, okay, that surround and protect the cell bodies. Okay, so notice this other type of cell here wrapping around the axon. And so what these other cells are doing is they're forming the myelin sheath. And that's the other type of neuroglial cell in the CNS, or, sorry, the PNS. It's called a Schwann cell or a neurolimocyte. So these do the same thing that the oligodendrocytes do, but these are in the PNS, okay? Oligodendrocytes are only in the CNS. So the myelin, again, does the same thing. It electrically insulates the axon. It will affect how the action potential travels. And that is actually what you see here, okay? So this is a myelinated axon. And these actually are, each of these is a neurolimocyte or a Schwann cell wrapped around the axon. So this is a separate Schwann cell here. Here's another one. This would be part of another one, okay, forming the myelin sheath. So let's take a look. Whether we're talking about the central or the peripheral nervous system, again, the myelin sheath is going to look the same. And this is what it would look like out in the PNS. Okay, we have a multipolar neuron. These are all individual Schwann cells wrapping around the axon. And again, just to compare, so this would be the myelinated axons in the central nervous system versus what it would look like in the peripheral. It's basically the same, just a different type of cell forms the myelin sheath. Okay, so now notice that in between these little sections of myelin, we have these spots where the axon is bare, okay, where it's not covered with the myelin. These are called neurofibril nodes, also referred to as the nodes of Ranvier. This is some French person who studied neurons. Okay, so same thing here, neurofibril nodes or nodes of Ranvier. These are important because this is the only part of the axon that can conduct the action potential. Okay, so this is the only area where that electrical current can travel across the axon. So this is how the myelin sheath affects the action potential. Okay. So let's take a quick look here. And we are almost finished, you guys, so just try to focus for a few more minutes here. So this is a little section of axon. Notice this is a neurolimocyte or Schwann cell. You can see here how it's forming the myelin sheath. It's going to start wrapping around the axon. And so this is a little further along in that process. It's kind of wrapping around it. And then notice that what happens is its membrane, the membrane of the Schwann cell, just keeps wrapping around and around and around the axon. So that is really the actual myelin sheath. It's all of these kind of layers of the membrane of the neurolimocyte. Okay, so this is the actual myelin sheath right here. Now notice what's left over outside of that. It's easier to see it here in this picture than in this one, so I'm going to use this picture here. This is the rest of the Schwann cell. This would be any leftover cytoplasm and also its nucleus, okay? So this part of the Schwann cell here is called the neurolemma, okay? This part of it is the actual myelin sheath. So these are both part of the Schwann cell, okay? It's all part of the Schwann cell. Okay, the myelin sheath and the neurolemma are both part of the Schwann cell. But I would like you to be able to identify those two different parts. So here, again, this is a little further along now. The myelin sheath is completely developed, and we still see outside of it the nucleus and some leftover cytoplasm of the Schwann cell. So again, this is what is called the neurolemma. This diagram doesn't label the neurolemma, but your book talks about it. Okay, and you can also see it if you look at the model. Okay, so you can see where the nucleus of the Schwann cell is out here. So this outer membrane, including the nucleus, is the neurolemma, and you can also see it in this cross section. Okay, so you should be able to identify the myelin sheath and the neurolemma outside of it. And these would be the nodes of Ranvier, or the neurofibril nodes between the Schwann cells. Okay. You guys have any questions on that? Okay, so let's take a quick look here at a myelinated axon and look at how the action potential is conducted. And you will see that there are these two different types of conduction. Okay, and oops, I think we have to go back to this picture. Let's use this one. Okay, so now we said that the action potential begins here. This is the axon hillock, where the action potential is going to start. Okay, so because the action potential can only be transmitted at these nodes of Ranvier, or neurofibril nodes, the action potential, again, begins here, and then it will kind of, in a way, it kind of jumps to that next node of Ranvier. From there, it will jump to the next node. From there, it jumps to the next node. Okay, so it has to skip over these areas of myelin. It seems like maybe that would slow the action potential down. It actually makes it travel much, much faster because it doesn't have to be transmitted along the entire axon. Instead, it gets to skip over most of the axon. So this actually causes the action potential to travel about 100 meters per second. Roughly 100 meters per second, so very, very fast. This is what's called saltatory conduction. Okay, and you guys will talk about this again when you take physiology. Saltaire means to jump. Okay, and so saltatory conduction is the type of conduction we see in a myelinated axon, where the action potential is kind of jumping or skipping along the axon. And again, it will go roughly 100 meters per second. Now, in unmyelinated axons, and we do have some unmyelinated axons in the body, the action potential travels the entire length of the axon. It's called continuous conduction, and it's still fast. It's going to travel about 1 meter per second, but it's much slower than in a myelinated axon. Okay, so I'd like you to know about these two different types of conduction and whether they take place in myelinated or unmyelinated axons. So you can see how in people who have multiple sclerosis, if their myelin sheath is breaking down, it will affect how the action potential travels, and it can actually prevent the action potential from being transmitted at all. Okay. Questions on any of that? Okay guys, so there's one more thing in chapter 14 that's easy, but let's take a break first. Let's come back at 9 o'clock. We'll finish this up. Okay, and you can look at some neurons